Hello, my name is Yana Wu, and the title of my project is Exploring the Therapeutic Potential of Albiflorin in Alzheimer's Disease. Alzheimer's disease, the sixth leading cause of death in the United States, is a progressive brain disorder in which the brain slowly deteriorates through neuronal death. One of the main lesions or biomarkers of Alzheimer's disease is the formation of beta amyloid plaques through the agglomeration of a misfolded sequence of the amyloid precursor protein. In affected brains, amyloid precursor protein is irregularly cut, forming beta amyloid that cover the synapses of neurons, leading to neuronal death. Following heat shock or cerebral ischemia, the brain produces heat shock proteins that act as chaperone proteins to refold these misfolded proteins. In normal brains, heat shock proteins are found at extremely low levels. This study focuses on inducing heat shock proteins through the treatment of a monoterpene glycoside, albiflorin, for the purpose as serving as a therapeutic treatment for Alzheimer's disease by targeting beta amyloid aggregations. The data obtained via cell viability tests, ELISAs, immunofluorescence, and confocal imaging suggests that the albiflorin treatment of 20 micromolar significantly increased the cell proliferation of human neuroblastoma SHSY5Y cells. Additionally, albiflorin treatments of 2 micromolar and 20 micromolar significantly induced heat shock protein 70 in the absence of heat shock, as well as changed the intracellular localization of the protein, thus highlighting the possible therapeutic effects for Alzheimer's disease. As seen in Figure 1, the treatment of albiflorin resulted in a phasic response. At the concentration of 20 micromolar, the treatment of albiflorin increased the percent viability in comparison to the control group. The results from the ELISA assay suggest that albiflorin treatments of 2 and 20 micromolar induces the production of heat shock protein 70 in the absence of heat shock. As seen in Figure 2, the treatments of 2 micromolar and 20 micromolar both increase the production of heat shock protein 70 as compared to both the control and solvent control with statistical significance. In order to view this expression, immunofluorescence using the laser scanning confocal microscope was used. As seen in Figure A1 and A2, in the heat shock group tested with 200 millimolar of ethanol, heat shock protein 70 is nuclear. However, as seen in figures B1 to C1, heat shock protein 70 is perinuclear when the cells were treated with albiflorin, as the proteins are more present in the projections of the cells. As the movement of heat shock proteins towards the projections of the cells, this may hint at a possible treatment of Alzheimer's disease, as the plaques often congregate at the projections between neurons.